Okay, let's continue talking about 7.9 and phylogeny. So we've said now we use DNA sequences. We actually can use DNA, RNA, and protein sequences to help us understand relationship among species. So we take this in addition to anatomical comparisons. It's not like all of a sudden we figured out DNA and we got rid of all those altogether, but we use them to look for similarities and for differences in those sequences to help us make things kind of less ambiguous and more objective, less about what we think, and maybe just provide us another piece of evidence. It's now really fast. We have lots of DNA sequences from many organisms, and the more we get, the better we can actually predict these relationships. You can look here quickly. Here's DNA and RNA base pairs in this bank, right? Like, what is the information that we know? And it has really skyrocketed. This only goes to 2011, but you can imagine it's even continued on from there.
So here's the amount of sequence you can generate for $1. Now, you have to pay more than $1, obviously. But if you reduce it to the cost of $1, this is how many bases you can sequence as a function of time. You notice how the curve is going exponential? In just the last 10 years, there's been a 50,000-fold improvement in how much sequence you can do for $1, which is unbelievable because nothing in technology changes by 50,000-fold in 5 years. Not semiconductors, not computers, storage, data systems, none of that stuff. So we're really on a different trajectory here. And what does that mean? It means we can view everything as sequence. Because now we have the possibility of literally sequencing everything.
So how are DNA sequences used? Well, evolutionary related organisms share some sort of common ancestor. So the idea is that they have some sort of ancestral DNA in common. As organisms evolve and diverge, their DNA sequences accumulate differences, and we know that these are called mutations.
How do we do this? How are we going to use this information from the mitochondrial genome, for example, to reconstruct human origins? Well, one thing we can do is simply look at the mutations that have occurred over time. So if you look at the sequence on the top, which represents a common ancestor, their sequence, and let's say that in the lineage leading to population A, there was a mutation on the left-hand side from T to A, it's highlighted in red, and then there was another mutation from C to G. And now everybody in population A has the A and the G nucleotides. Population B, they by contrast had this mutation from an A to a T, and everybody now has the T. So if you were to construct a phylogenetic tree, you would have all the individuals from population B that share that change are going to cluster together. All the other individuals are going to cluster together. Now,
So in order to figure out what they share, we have to compare the sequences. And to do that, we actually have to align the sequences. So we look at things that are evolutionarily, we think, related, or homologous sequences, things that relate to each other. And we basically go through and we put them all next to each other, we run them back and forth, and we see like, oh, where are the highlights? Where are the most commonalities? And we just try and maximize the number of matches in a sequence by changing the position in them. Each line of DNA has a sequence and a position, and a little star would indicate, that all the letters in that column match up. So you kind of just like run them against each other. Look, it's kind of highlighting, oh, here are matches, right? And we're going to try and move it back and forth essentially to get the most matches that you can. We as humans don't do this. We have computers that do this and try and find that high match sequences together, and then we compare them, look at how they're the same or different. More distantly related species have had more time to change since they shared some sort of common ancestor. So we say that because they have had more time to change, they have more mutations. They are more different. Those mutations have had more time to accumulate. Closely related species have had less time to accumulate mutations because they have a common ancestor that's closer, so their DNA sequence would have more in common. We kind of refer to this as the molecular clock and figuring that out. We can have single nucleotide polymorphism where we have just one mutation, right? It's a pretty common mutation that can be used to track common ancestry. It's just a change in one base pair so that the alignments don't match. Maybe we just changed that T or to a G, right? That's a single nucleotide polymorphism. Think of it as a single point mutation, and that might be all we see in that change over time. We can have indels. We refer to them as insertions or deletions. One or more nucleotide pairs in the DNA strand is lost or gained, and it gets represented by a dash or a gap when I'm looking at those two comparisons. Again, it's called an indel. It can be an either insertion or deletion. So say we could get everything to match except maybe this one place. That would be a good indication that that's what we saw happen. We can actually compare many sequences at one time. It's not just that we can run two sequences of a gene across each other. We can look at it through many different species at many different times, run it back and forth, look for kind of similarities, and maybe we would even see more than one change over time between the two. Maybe it's even in different places, but that helps us gather information as well. And then phylogenetic tree from our DNA. There's several different ways to create a phylogenetic tree. One method finds the two most similar and therefore likely closely related sequences, aligns them, and then works backwards to find less closely related ones.
Every single individual mutation, we can just summarize based on all the diversity how close two lineages or two sequences are. So for example, in sequence 1 and 2, you can see in red that they differ at 3 out of a total of 20 nucleotides. Okay, so that's about a 15% difference. And you can see this little matrix, we're comparing now sequence 1 and sequence 2 in red, 15% difference. Now if we compare sequence 3 and 4, they differ at 2 out of 20, or 10% difference. So now let's make a topology. And we can see that we're going to put 1 and 2 closer together, and we're going to put 3 and 4 closer together, and we can see that if you look at that matrix, 1 and 3 are much more distant from each other.